Okay, this is our second video focusing on the absorption of macromolecules in the small intestine. And so the first video was carbohydrates and proteins, and then this last one we'll talk about lipids and nucleic acids, and then other substances that get absorbed in the small intestines. So let's take a look at lipids. Lipids are hydrophobic. That means they do not like water. And so that quality makes their digestion a little bit more complicated than carbohydrates and proteins. Basically, for both carbohydrates and proteins, we were just taking the big chain and breaking up into smaller pieces, and then finally those smallest pieces were absorbed in the small intestines. Again, because water is hydrophobic, that makes it a little bit more complicated. So we've got a few different lipases. Those are just fat digesting enzymes. We see one in the saliva, which is active in the mouth, but then becomes more active in the stomach when it is joined with another lipase, gastric lipase. And so 10 to 15% of lipids are digested before reaching the small intestine. Again, there's quite a few lipases, but they are all breaking down of lipids. Pancreatic lipase is emptied into the small intestine, which digests most of the fats. So the fat enters the duodenum as large globules, and it's only exposed to lipase at their surface. And those globules are then broken down into smaller little droplets by components of bile. And so there's a picture in your book that shows the bile acids kind of surrounding these little droplets. They basically coat the acids, and it keeps them broken apart. And so that, again, increases the surface area for the enzymes to kind of do their job. So there's plenty of pancreatic lipase in the small intestines after a meal. It's able to digest the average daily fat intake in as little as 1 to 2 minutes. So plenty of lipase going around. When the lipase acts on a triglyceride, it removes the triglyceride has 3 fatty acids, hence the name tri. And so the lipase removes the first and third, and that from the glycerol backbone, and it leaves the middle one. So what's left is a glycerol and then 1 fatty acid. And so that product there is 2 free fatty acids, and then a monoglyceride, which is still a glycerol and 1 fatty acid joined together. And so then those smaller particles are able to be absorbed in droplets within the bile called micelles. And so they're made in the liver, and you can see a close-up. I don't know that there's a picture here. Oh yeah, that's not a great picture. There's a better picture in your book that gives you kind of a close-up. Again, it's still breaking a big lipid down into smaller pieces, but it goes into a little bit more detail about that the bile acids kind of have to wrap around the free fatty acid, and just kind of how that process happens. So we would expect to see, this talks about their arrangement. I'm not really picky about that, but the bile, phospholipids, and the cholesterol are able to diffuse into the center of the micelle to form the core. Now again, it just, in your book, it just kind of looks like a yellow circle. I'm actually going to go forward to these pictures. Again, there's a bigger picture in your book. This is kind of a teeny tiny one. But it just means that it has the bile, I'm going to go back. It has the coating of the bile on top of this little droplet. And that's what a micelle is. So these micelles are able to go down the bile duct into the duodenum, and then they are absorbing these fat-soluble vitamins, cholesterol, and the free fatty acids, and the monoglyceride. And so once the micelles are able to be in the small intestines, they transport the lipids, they kind of latch onto the lipids and bring them to the surface of the absorptive cell in the intestines. So the lipid leave the micelles and then diffuse through the cell membrane, and the micelles are reused. So they're able to pick up another lipid, transport it to the intestinal cell, and recycle that process, or are reused in that process. So don't get confused about the micelles. I think they're weird little things. But they are basically bringing the free fatty acids and the monoglycerides to the surface of the intestinal cell. So taking it to that brush border. And that's what this picture is, and this picture is much bigger in your book. So there's little micelles, and they're bringing the free fatty acids or the monoglycerides to the absorptive cell of the small intestines. And then it talks about them making that kind of these combined lipids and proteins. I'm not really picky that you call them chylomicrons. I think a lipoprotein makes way more sense if it's a lipid and a protein. But basically, once the micelles are bringing the fatty acids and the monoglycerides into the absorptive cell, they basically reform back into triglycerides, and then the triglycerides can combine with the protein to make these lipoproteins. And so then they're packaged in vesicles that go to the surface of the cell, and then they are released. They are released into lymph, and then eventually the lymph vessels, we know kind of the system of that, and then eventually these lipoproteins will re-enter the bloodstream. And so here's a better picture of once the lipoprotein is in the small intestinal cell, it's released from a vesicle, and so there's a vesicle going towards the outer boundary of the cell, and then it's released through exocytosis, and then it sneaks into the lymph through those little gap junctions. Okay, so the last little bit that we have to talk about, our last macromolecule, is nucleic acids. Now you notice this is so much shorter than any of the other types. The nucleic acids are so much easier, and they're much smaller in quantity as far as how many nucleic acids that we are absorbing. So we have enzymes to break apart the DNA. They are called nucleases, and so they break apart the DNA or RNA into nucleotides. And then we have more enzymes at the brush border that split the nucleotide into its three parts: the phosphate, the sugar, and the nitrogenous base. Now, all of this should be review from AMP1. You should know that the nucleic acid is made of nucleotides, and that the nucleotides are made of phosphates, sugars, and bases. But that's as short as that part is. It's very, very simple. Just an enzyme for each one. Okay, and then vitamins are actually able to be absorbed without any change to their structure before they're absorbed. So they are absorbed unchanged. This talks about that fat-soluble vitamins are absorbed with other lipids. If they're not, if no fat-containing food, then they are not absorbed at all. It talks about water-soluble vitamins B and C are just absorbed by simple diffusion. Okay, let's go through minerals absorbed all along the small intestine, not just at one section, so all the way through. I'm not really picky about really memorizing all of these. Just know that we need to be absorbing sodium and chloride. You've learned other places in our body where we need those ions, so it is important that they are absorbed. And then this talks about specifically calcium is absorbed throughout the intestines. It's absorbed by active transport in the duodenum, and then diffusion in the jejunum and the ileum. But again, sodium, calcium, and chlorine are minerals that your body is going to absorb. I'm going to jump past here. Water is absorbed in the digestive tract, and we're going to actually talk about in the last video about the large intestines exactly where. But let's talk about it here for just a second before we go on to the last video. The digestive tract receives 9 liters of water a day. So some of that comes from food, some from drink, obviously, and then all of these secretions that we've been talking about are mostly water. So some of that, most of it, is absorbed by the small intestines, 8 liters, and then in the other two video, or the last video, we'll talk about the large intestine and defecation. So for the small intestine, the water is absorbed by osmosis. So once the absorption of salts and nutrients, then the water follows that just by osmosis. So the sugar or salt content of the absorptive cells in the small intestine is then going to pull water to follow it. And that's, again, going back to A&P1 and just studying up on osmosis.